Thanks so much for the introduction. We're really excited to be here to kick off the first MPG primer of the season. And we're going to be talking about genetic diversity in human pluripotent stem cell models. So we'll start with a brief history of stem cells. We thought it would be interesting just to give an overview to demonstrate the trajectory of the field. We're then going to talk about genetic diversity in some of the large stem cell collections, when we think this genetic diversity may matter experimentally and when it may not. Some of the challenges that we face in terms of diversifying the stem cell collections and really where we'd like to see things go in the future. So please, we'd love for this to be more of a discussion, so feel free to interject and ask questions as we go along. So to make sure we're all on the same page, what is a stem cell? We're talking about cells that have two key properties. The first property is self-renewal, so they're able to make indefinite copies of themselves. And the second property is differentiation, so they're able to generate more differentiated progeny. And there are many different types of stem cells. Today, we're going to be talking specifically about pluripotent stem cells, which means that they can generate all of the differentiated cells in the body. So the term stem cell actually first appeared in the scientific literature in 1868, and it's credited to a German scientist, artist, and philosopher named Ernst Haeckel. And he said the name stem cell seems to be the most simple and appropriate one because all other cells stem from it. And because it is in its most literal sense, the stem father as well as the stem mother of all the countless generations of cells of which the multicellular organism is later composed. And so it was recognized quite early that there were cells that had the capacity to generate all of the diverse lineages of an organism. And you can actually still buy some of his books on Amazon. He made some really interesting illustrations of art forms in nature. So for brevity, we're going to move forward about 100 years to 1981. When work from Martin Evans, Matthew Kaufman, and Gail Martin derived the first mouse embryonic stem cells from blastocysts. So these are a type of pluripotent stem cell. They're capable of generating all of the lineages in the body. And this involves taking a pre-implantation blastocyst, isolating something called the inner cell mass, or the ICM, and then being able to dissociate and propagate those cells indefinitely in vitro. And this was really essential work for future generation of mouse knockouts. And it was also proof of concept that something like this could potentially be done in human as well. And shown below is just a generic stem cell colony. This is what they look like when they grow in vitro. It's this ball of cells here in the center. A few years later, Andrew Lasser and Hal Weintraub at the Hutch discovered that you could directly convert rodent fibroblasts into another cell type called myoblasts using a single transcription factor, MyoD. And this was really an early conceptual advance because it showed an unexpected degree of plasticity in cell fate. So previously, you would think of cells differentiating in one direction and that being a one-way street. And what they demonstrated is that these cellular states could be manipulated. This is a process called transdifferentiation, and it's something that we use in the lab today. So this is a really critical advance. One of the limitations is the cell number, because with transdifferentiation, you're not transiting through an expansion phase. In 1996, I'm sure many of you are familiar with Dolly the sheep. This was the first mammal born from cloning, and it was work from Ian Wilmot, who unfortunately just passed away earlier this week. The technique is something called somatic cell nuclear transfer. It's very inefficient. I think it took 277 tries before they were able to get Dolly. And essentially, it involves taking a cell from the body with the desired genotype, popping out the nucleus, and then putting it into a denucleated egg. Since Dolly, there have been many other mammals born through somatic cell nuclear transfer. Many different mammals have been cloned. And obviously, it's led to controversy about the potential applications of this technology in humans. A couple of years later, Jamie Thompson and Jeffrey Jones were able to isolate the first human embryonic stem cells from blastocysts. This is a line called H1. It's still standardly used in the lab. And I think it's noteworthy that this occurred a full 17 years after mouse. So it wasn't a straightforward leap from being able to do this in mouse to being able to do this in human. But it involves the same principles of taking a pre-implantation blastocyst, isolating that inner cell mass, and then being able to continually propagate those cells in vitro. Human embryonic stem cells are tightly regulated, and they do come from embryos that are left over from in vitro fertilization clinics. And this involves the destruction of a human embryo. So there have always been ethical considerations around their use, although they've led to some really important discoveries in science. And I think an important limitation here is that there's really no human patient. So if you want to make those interesting connections between a patient phenotype and the human genotype, you would not be able to do this with embryonic stem cells. These two problems were solved a couple of years later from work by Takahashi and Yamanaka. Yamanaka subsequently went on to win the Nobel Prize. And that was the first generation of induced pluripotent stem cells, which is primarily what we're going to be talking about today. These are cells that are functionally equivalent to the embryonic stem cells, but they're generated by converting a somatic cell type to the pluripotent state. They first did this in mouse fibroblasts using a retroviral approach. When this paper was published, there was a lot of excitement in the field, and I think a fair bit of skepticism about whether this was actually technically possible. But the technique was really quickly recapitulated by labs around the world. 1 year later, they did the same trick in human. A couple of years later, there was the first FDA-approved trial for a therapy based on embryonic stem cells. And in 2014, the first clinical trials using iPS cell-derived cells, in this case, retinal pigment epithelial cells. So this was just an astonishing speed to go from the inception of the model system to their use in clinical trials. And just this year, the labs of Magdalena Zeneca-Götz and Jakob Hanne succeeded in generating the first post-implantation human embryo models. So these include both the embryonic as well as the extra embryonic tissue. So shown in blue is the embryonic material, in yellow, the yolk sac, and in pink, the placenta. So these models can be used to study the very early stages of development, and they're not taken beyond day 14. There's a day 14 rule in the field. So we walked through this history really to show how rapidly this field is moving and then some of the different directions that it's going. Because we're going to be talking a lot about iPS cells today, we just wanted to go over what that means. iPS cells are derived from somatic cell types. Anyone could be a donor. You can give blood or fibroblasts or other cell types to then be converted into pluripotent cells. You take that biopsy material and transiently introduce different reprogramming factors that push these cells down to a pluripotent state. Once you have those induced pluripotent cells, they can again be propagated indefinitely in vitro, and they can also be used to generate, in theory, any different cell type in the body. So the promise of this technology is really for things like cell replacement therapy, to understand the relationship between a variant and function, pharmacogenetic testing, et cetera. They are really highly scalable, which is very useful in the lab. In theory, they can also be used for autologous transplantation, and you can have a direct link to a patient phenotype, which is really critical. So I'm now going to turn things over to Ralda, who's going to talk about some of the efforts to really expand these resources into larger banks and how that dovetails with some of the human genomic efforts.
Hi, thanks so much, Lindy. So yeah, I think with kind of following this wonderful introduction from Lindy, one thing to note here that I hope is quite relevant for the MPG community is that breakthroughs in the stem cell reprogramming field really went hand in hand with breakthroughs in human genomics over the last 20 to 25 years. And I hope you can appreciate this from this timeline here, where, again, thinking back to what Lindy just told us, the derivation of the first human embryonic stem cell line happened in 1998. Shortly after that, at the NIH, an embryonic stem cell registry was created a couple of years later, and that shortly after that, the first human genome project was completed. Then after that, the first human induced pluripotent stem cell was generated. The next thing is that the 1000 genomes project was completed in 2008. A few years later, large-scale iPSC repositories were launched. And then again, the year after that, large-scale genetic studies such as PubMed, H3 Africa were initiated. Enrollment started for all of us in 2018, and so on and so forth. So from this, really like the fact that like the revolutions in these two fields were happening in parallel, this then provides unique opportunities to be able to really understand how human genetic variation might shape human traits and disorders. And we can do that in a dish in the lab from human cells. However, despite all of these advances, one thing that really was kind of people came to appreciate in the human genomics field is that a lot of participants in these genetic studies are historically not representative of global populations. So you can appreciate here, and I'm sure this has been discussed in this venue many times before, that most of the GWAS participants, for example, are of European ancestry. And to rectify this, many efforts have been launched to prioritize the inclusion of underrepresented participants in human genomics research. And some of these initiatives are highlighted here on this map. And so why this is important there has been, again, a lot of work, many of it from Brodies themselves, that have highlighted that when, for example, polygenic risk scores, when genetic studies are kind of underrepresented populations, the resulting polygenic risk scores, for example, are not representative of these populations. And so then these are less accurate in a clinical setting, and this can in turn exacerbate health disparities. Conversely, when we do take like genetic diversity and different ancestries into account into the polygenic risk score approach, that can greatly improve risk prediction. And an example is shown here again from the Broad in the context of coronary artery disease. So as these genomic studies diversify, there will be many more alleles that are differentially enriched in different populations. And there is then a need to be able to have experimental models that can capture all of these risk variants and enable us to understand how they affect human traits and disorders. And as Lindy mentioned, human pluripotent stem cells can be obtained from many different people in any one geographical location, and they can be differentiated into disease-relevant cell types, and they can even be further engineered to harbor one or more genetic variants of interest. So to meet this need, there has been several large-scale stem cell repositories that emerged worldwide, and to ensure that there is a standardized way to collect and bank the stem cells, to reprogram them, store them, and distribute them. However, unfortunately, and this mirrors what we have seen in the kind of genetics field, the existing human pluripotent stem cell lines in these large banks are really not representative of global populations. So as you can see here, we've shown some of the largest repositories worldwide, and the majority of participants in these, like the people who donated these cells, are of European ancestry. And this is another example when we look at the human pluripotent stem cell registry that really catalogs all the stem cell lines in these banks, again, using standardized biological and legal agreement. Again, we can see that worldwide, 73% of human pluripotent stem cell donors are of European ancestry. However, there are still some local efforts that are starting to emerge at the national level that are really trying, with a focus on really providing healthcare to particular community, that are trying to bank and DPQC stem cell lines from specific populations. So this is happening mostly in Korea and Japan. And so the Korean stem cell bank here on the left, and right can be Procell and Sira in Japan. In the case of Sira and the Korean stem cell bank, they tried to really focus on a few cell lines. So the total number of cell lines is indicated here at the top. So you can see that here we're only talking about a handful of lines. But again, these have been kind of selected very carefully, for example, to try to identify donors of a specific haplotype, in this case, the HLA haplotype. And we'll talk a little bit more about this in a moment. Here at the Broad, we also have a stem cell resource that we've established, and that now consists of stem cell lines from over 800 unique donors that have been banked and sequenced. And the breakdown of the ancestry of these lines is shown here at the bottom. So you can, I hope, appreciate that it is quite similar to the breakdown in the US Census that is reported. However, we still have a majority of donors from European ancestry. So does it really matter in functional studies to have genetic diversity? Does such genetic diversity influence cellular phenotypes, specifically in the context of stem cell studies? And the answer is yes. There is an increasing body of evidence that supports the fact that we can see differences in cellular phenotypes based on the genetic background in human pluripotent stem cell models. Here we're highlighting this study from Helena Kilpinen and her colleagues, whereby they have used 301 individual cell lines derived from 301 individual healthy donors and derived iPSCs, and then profiled them in a variety of different ways. And what they found is that individual donor effects explain up to 26.3% of variance in gene expression. They further found that genetic differences between donors affected many cellular traits, including differentiation capacity and cellular morphology. And there are many other studies that further support this. So when does it actually matter in the lab whether or not we are using genetically diverse human pluripotent stem cell models? Excuse me. So there's one case that it may not matter whether or not the stem cell lines that we're using are genetically diverse. And this is in the case of monogenic diseases in a case control study, for example, not known to vary by ancestry. It is often the case that monogenic or Mendelian, in the context of monogenic or Mendelian disorders, there's a pathogenic variant that will lead to disease regardless of what population the variant is found in. However, when it likely matters is when we are thinking about population-specific risk variants, and that can even include Mendelian diseases, in the context of cell replacement therapies and drug safety and toxicity studies. And I'll give examples on each of these points. So starting off with population-specific variants, I want to highlight here this study, H3 Africa, that has generated whole genome sequencing of 426 individuals from 13 different African countries. And the study was able to actually identify 3 million new variants that have not been identified before. And these discoveries are very, very valuable in, like in the context of clinical interpretation of whether such variants may or may not be pathogenic. And so they've added a lot of knowledge from that perspective. But the study has also been able to identify many new disease-associated variants or rare variants. And such discovery from a somewhat small sample size, much smaller than what is typically done, and for example, European-based population has led to many more discoveries, again, because of many different reasons, including that African populations typically have shorter LD blocks. So again, there can be a lot of new discoveries that can come from studying different populations. Another area where it matters whether or not genetic diversity is captured in human pluripotent stem cell research is when we think about cell replacement therapy. And this is something Lindy had alluded to already. So in this context, people think about, for example, stem cell transplantation to replace a cell type that is no longer there or that is kind of affected by a specific disorder. And the gold standard in this approach is to take one's own cells and transplant them back into the body. So this is called autologous transplantation. However, it costs a lot of money and takes a lot of time. And so it's most of the time not realistic to do that. So another approach that the field has adopted to circumvent immune rejection is to match donors by HLA haplotypes. So, and in this approach, induced pluripotent stem cell lines are screened and identified from donors that are homozygous for a given HLA haplotype. So one can actually select cell lines, induced pluripotent stem cell lines that are homozygous for a number of HLA haplotypes in a way that you can capture the majority of a given population. In this case, only 10 iPSC lines would provide a representation or a match for 2/3 of the UK population. And a similar approach, and because of the value of this approach and kind of the interest of the field in cell replacement therapies, this is another study that is now based in Taiwan, where they have screened 1,000 cell lines from, or DNA from 1,000 participants in Taiwan, and they identified 13 donors that are homozygous for HLA haplotypes. From these samples from the blood, they've then derived induced pluripotent stem cell lines. And then they label these donors as super donors because only these 13 lines can represent 477 million people worldwide. So it provides a beneficial match for that many donors. And they even went further, and from these iPSCs, they've generated cardiomyocytes and neurons and performed toxicity screens, again, showcasing the value of this approach, even kind of going to a kind of more translational perspective. So this then brings me to my last point, is that another important area where we should think about genetic diversity is when thinking about drug safety and toxicity studies. So there is right now limited information about drug efficacy and safety in non-European populations. In fact, most clinical trials are often based in the UK and in Europe, and as a result, we often, again, don't know if individuals in other countries who are taking these drugs might or might not develop side effects. And in fact, it is often the case that individuals in different countries, for example, in South Africa, there has been a report on this, they might be hospitalized, and most of the cases are because of the drug that they're taking and instead of a disease they're sick from. And this here, this table shows that for many of the drugs, there are often ancestor-specific differences in how they respond to treatment. So while it's not, convenient to run clinical trials in many different countries and continents, it is often possible to take sample lines from any person in any geographical location, and then derive stem cells and then run toxicity and efficacy screens on these cells. All right, so I hope that we've convinced you that there are many advantages and actually a need, scientific as well as ethical, to diversify stem cell resources. However, we should keep in mind that this is not an easy task, and we are well aware of this. So first and foremost, all of these the recent effort around greater inclusivity, they exist in a historical background of a lot of discrimination, which have led to really mistrust of many in the scientific and medical establishments. So really we should, as a field, make conscious efforts to really increase engagement and make sure that in these approaches, they're not only beneficial to one party, but the benefits of the science and the work are really well explained to everyone without ambiguity, and they are also shared. And this can be done through, for example, scientific partnerships and et cetera. So this really then should shape how we think about obtaining or establishing studies of induced pluripotent stem cell lines, how we collect them, again, making sure that it is a collaboration as opposed to safari science, expanding collections locally, and then making sure that we also invest in training and capacity building and or expansion in underrepresented settings. Also, how the data is governed can be really challenging, obviously from a scientific perspective. The more data, the better. And we want this data to be publicly accessible to everyone. We want deep clinical phenotyping. We want to be able to share the data and the cell lines with anyone, anywhere in the world. However, it's also important to balance this with the need for to protect personal health information and donor privacy and the many rules and legislation that exists around this. The cost for using many different cell lines, diverse cell line, can be really, really high. And Lindy will tell us more about this in a moment. And finally, the way race, ethnicity, and ancestry data is currently reported, cataloged, and linked to human pluripotent stem cells is also problematic. And again, Lindy will tell us more about this.
We thought it would be helpful to go through examples of two of the challenges, starting with the really exceedingly practical example of cost. For those of you who don't work with stem cell models, these are incredibly expensive, both to generate and use in the laboratory. So if you wanted to generate a research-grade iPS cell line, that would run you in the ballpark of $10,000 to $25,000 for a single line for the reprogramming and then the validation of that line to establish the resource. That means if you have a classic 3-case, 3-control study, which I think everyone in the field would agree is very underpowered, you would be looking at $60,000 to $150,000 for that. If you wanted to get into the business of generating clinical grade iPS cell lines, the cost goes up substantially. And even if you're able to buy these cellular resources from established banks and you don't have to go through the reprogramming process yourself, carrying stem cells in the lab is quite costly. We tend to estimate around $3,000 to $5,000 per researcher per month in the lab for people who work with stem cells. And that's just to cover the media, the supplements, the growth factors, the substrates, and everything that goes into the wet lab work of just propagating those lines. And obviously, if you're doing more complex differentiation protocols, making 3-dimensional structures, the cost will go up from there. There are also real personnel costs. So these lines are not trivial to work with in the lab. If you cultured something like cancer cell lines, stem cell lines require a lot more care and attention. So while it does vary somewhat any individual typically carries between 4 to 6 independent iPS cell lines in the laboratory at any given time, just because of the maintenance required for the cells. So as we talk about expanding the genetic diversity, including things like replication studies or building out these larger multi-ancestry cohorts of stem cell lines, that's going to be out of reach in a cost perspective for a majority of labs, both to generate the stem cell lines, but also just to integrate those lines. And we're currently not aware of any funding mechanisms that would specifically support increasing the diversity and the number of lines that are used in these types of studies. So I think we have to be really thoughtful about how we move forward with expanding these lines in the lab. There are also considerations around how race, ethnicity, and ancestry data is linked to the stem cell resources. So taking you back to this chart that Ralda showed, this was actually really difficult for us to generate because when we look in the cell banks, the labels are not what appear on the right. So they're not broken into these different categories. And we had a lot of Zoom conversations trying to figure out what cell banks actually meant when they were talking about race, ethnicity, or ancestry. And that's not great. So just to give you some examples of how individuals of European and Asian ancestries are reported in the stem cell banks on the left, and how they may be reported in human genomic studies on the right. And what you can see is that there's, for the stem cell banks, no concordance between the banks or with the human genomic studies. So it can be very difficult if you read a study from the human genomics field and identify some really interesting variant in the population that you want to then interrogate functionally, to be able to match lines from the same genetic ancestry because of these differences in nomenclature. And a lot of that is historic and how the collections were sourced. We are only aware of one bank that actually uses genetically inferred ancestry, and that's the UK-based TIPSCI, where they give information about the predicted population and they break down into the major categories. So that's very useful. But the vast majority of the other banks use self-reported racial or ethnic identity. And this is just an example of all of the different terms that could be used to describe a cell line from one of the banks. This is Y cell. So it's important to note that the racial or ethnic identity is not the same as the underlying genetic ancestry, and those two pieces of information may or may not be concordant. But the only piece of information we're really getting with the stem cell lines is the racial or ethnic identity. It's also from self-reports. And so we wanted to think a little bit about whether that would be accurate, because again, these lines are really expensive to work with, and it's devastating to find out down the road that what you have been working with is not what you thought you had. So we looked into a study. This was looking at the reliability of self-reporting in the context specifically of carrier screening. It probably won't come as a surprise to this audience, but for individuals who self-reported with a single identity, when they then compared the major imputed ancestry, this was highly accurate for individuals of European descent, but things tended to break down in other categories. They had the interesting example of a breakdown for individuals identifying as East Asian, Southeast Asian, and South Asian. And they found that there was confusion about the terms for participants. So then when they were checking boxes on a form, they were not checking the right box because they did not understand what the terms meant. But when there was a one-on-one interview to ascertain this information, the accuracy went up. For individuals in the United States who self-reported as African, not unexpectedly, there is a range of the fraction of African ancestry versus European ancestry, and more dramatic range for individuals who report as Latin American. So I think the bottom line from this is just to emphasize that stem cell labs are generally not well positioned to determine the genetic ancestry of the lines that we're working with. And so we really rely on the information that's coming from the cell bank and rely on that information being accurate, because these are really lines that can live forever in the lab. And so what we'd love to see, and Ralda will talk about this a little bit at the end, is at minimum, more consistency in the nomenclature between the genomics community and the stem cell community, because that way we can better match resources to follow up functionally. We would love for the resources in the banks to have information about the genetically inferred ancestry, not just the self-reported racial or ethnic identity, but they're both useful pieces of information, but they're different pieces of information. And in an ideal scenario, when information or when samples are collected for human genomic research, we would love to have iPS cells taken into consideration. So you have cellular resources connected directly to the genomic research. So finally, we just wanted to talk briefly about how we think about building out stem cell cohorts and following up functionally on studies that come out of the genomics field, and where we might run into issues in trying to diversify those resources. So if we go back to this chart again, the Y cell collection and the SERM collection, between them, they have over 4,000 lines. They're quite large. And you can see that while the majority are derived from individuals of European descent, we do have these other ancestries represented. So could we actually go into those banks and build out the kinds of cohorts that we might want to? So we'll just walk through two theoretical examples here. This is a paper from Nature Genetics where they identified a candidate effector gene at a COVID-19 risk locus. And in the study, they reported that risk variants of the locus were carried by individuals of South Asian ancestry. So if we read this study and we were inspired to follow up on those variants, we might want to build out a stem cell cohort that has individuals of South Asian ancestry. And could we actually do that? So we went to the major repositories, first Coriell. So we wanted to look for stem cell lines. We don't want them to be disease affected. And we picked all the categories that we thought could potentially represent that ancestry. And that gave us 3 lines and none appeared to be the correct ancestry. We then went to SERM collection where we identified 4 lines and Y cell where we identified 6. So from all of the major US repositories, which contain thousands of lines, we were only able to identify 10 individuals that were likely to be of South Asian ancestry to build out our cohort. And obviously that is very underpowered. And when you consider that variants are expressed in a fraction of the population, we immediately run into a roadblock here, just looking in an unaffected population. We also obviously want to study disease. So if you take the example of this paper that came out recently, looking at the association of an African ancestry-specific ApoE missense variant that confers risk of Alzheimer's disease. So if we read this study and it inspires us to build out an iPSL line cohort to study Alzheimer's disease, and we want to have representation of different genetic ancestries, can we do that? Alzheimer's disease is obviously very prevalent. So we go to the major banks and we're able to identify 83 Alzheimer's affected iPSL lines. And just to get to the point here, when we look at the underlying information about genetic ancestry, we found no lines from individuals of Asian descent, 2 lines from individuals likely of African descent, 9 Hispanic or Latino, 4 unreported, and the vast majority of European descent. So we would immediately not be able to build out a cohort to study a disease as prevalent as Alzheimer's that has representation from different genetic ancestries. So these are the points where we're running into problems in terms of following up on studies functionally. And that also really highlights the ways in which we need to build out cohorts in different directions in terms of unaffected populations and specific diseases where increasing diversity of donors would be really the most impactful. So with this, I'm going to turn it over to Ralda, who's going to talk about some of the directions we really want to see this move in the future.
May I ask one question from online while you transition? Is your impression that the costs associated with human pluripotent stem cells are decreasing overall? And what you think the kind of barriers are to helping bring those costs down?
So I would say from the wet lab perspective, we have not seen the costs decrease. When you buy a $400 bottle of media, it gets you 500 mils, and you can calculate how much each mil of media costs you. And that doesn't include all of the growth factors and supplements and the sterile technique that's required. All of those costs, if anything, I think that they've gone up. So definitely what we ballpark for spending in the lab is increasing. In terms of the reprogramming itself, I think that those costs can come down, particularly with automation. There are some platforms that are attempting to automate the reprogramming process, both to increase the reproducibility and potentially also to bring down the cost associated with reprogramming itself. Let me think.
Yeah, maybe just kind of adding a little different perspective is that when thinking about generating one iPSC lines, if you really want to start from scratch, then you have to figure out a way to fund, unless you're starting hand in hand with the genetic studies, you have to kind of recontact specific patients if you're selecting based on a genetic study. And then, then you have to fund like clinicians or nurses, et cetera, to get the sample. Then the reprogramming can happen after that, right? So it's a lot of things, and these costs are really not going to go down. If anything, they would go up. With inflation, Lindy talked about the cost of reagents, and that has really gone up by more than 30%, since the pandemic with all of the inflation. So the other aspect of this is that even though specific maybe reprogramming facilities, their costs and with automation are going down. Actually, the Harvard IPS core just released new prices, and if anything, they've gone up, right? Kind of the other aspect of this is that the NIH is increasingly putting pressure because a lot of these stem cell derivation efforts are done ad hoc in this facility or the other, in that lab or the other. And so the NIH is starting to crack down for standardization. So, and requiring that you deposit the lines in their own repository, while they take $2,000 or $3,000 to intake new lines, right? So if you're writing a grant, you want to propose reprogramming or sharing a new cell line. So then again, these costs are going down. So long story short, not coming down anytime soon. Yeah, but there is a need for that, right? So anyway. So just wrapping up, and again, as Lindy said at the beginning, this is really meant, we'd love to kind of discuss around these and think together with this community about some of these points that, we hope, again, are not only relevant for biologists, but also geneticists who are eager to have kind of the interpretation of human genetic variation come more to fruition, especially in a disease setting. So what would an ideal scenario look like? Again, like Lindsay mentioned, in an ideal world, we would the stem cell derivation efforts would be accounted for at the very beginning when a specific genetic study is established, right? These participants would be given the option to consent to IPS line derivation. And that way, from the beginning, all of the metadata, anything associated with the samples, the same blood sample that is used to extract DNA for genetic studies can also be used, processed differently. The PBMCs can extract it and be used to derive stem cells, right? So there are large efforts that have done this. For example, the NeuroDev initiative here at the Broad, they've done this already. The samples are sitting in a freezer. The kind of blood cells are sitting in a freezer somewhere in the NIH repository waiting to be reprogrammed, right? So again, this is a clear way to link clinical phenotyping data, any other metadata with the cellular resource. Also thinking about the shareability and access of both the cellular resource and the derived genetic data or any other data that is generated, making sure that these resources are accessible really in a collaborative way. And this is, the idea of scientific partnerships. If we're thinking about genetic diversity, right, we often have to go and kind of derive the stem cells from more than one population, more than one source, but we also have to think about how to do this in a selfless or not very selfish way, right? So again, establishing these scientific collaborations that can benefit everyone. And then also local collections can be really a great source of iPSC materials. And then one important aspect to invest in upfront is really education, training and capacity building or capacity expansion in underrepresented population to make sure that individuals in these populations really understand what can be done with the stem cell lines, and they have access to the resources that are needed to be able to use these lines and interpret the resulting data. When we think about how to kind of properly catalog the stem cells that we have, so it is, again, important here to make sure that we are using, especially as one thing that is coming down is actually the cost of genetic sequencing, right? Data generation that is coming down, so that we think that this is going to be more and more common in the future. However, again, not necessarily accessible to all labs, but as the field is moving towards these large repository, then being able to make use of this data and use genetically inferred ancestry that would be paired with race and ethnicity as opposed to just self-reported, for example, will greatly increase the value of these lines and their reliable use in downstream studies. It's important that the same clear language is used in cell banks as in genomic studies. Again, just to make sure that the reliability of this information is, again, robust and translatable. And also, there needs to be clear consents and rules about the use of the cell lines and the data. In what context, can they be shared or not? And again, this is something that researchers often think about like the last minute before publication, but it's actually, then you can end up with cell lines where the data can't be shared or the cell lines actually can't be shared. You get in trouble with the journal and with your funding agency. So this is something that really should be thought about upfront. And finally, at the level of single labs, it is really critical to have funding from a federal source to support these initiatives, right? Because again, like we just discussed, it's extremely costly to diversify stem cell lines and use many stem cell lines. It's often inaccessible for many different labs. However, if there is a funding source that people can reach out to and get extra support for this, that could be really, really beneficial and make it much more accessible from also the clear and accurate language that describes not only the cohorts that are being used, but also why they were used and the possible role of ancestry in biological findings, right? This can be, it's a point that can be open to misinterpretation. And we've all read, right, in the news media, how, any conclusions that are made not very carefully, can really lead to a lot of controversy and misunderstanding. So it's really important to be very, very careful about the conclusions that one draws and what language is used to describe this. So with this, we really would like to open it up to discussion. But before this, I would like to thank you all for your attention, but also thank specifically Dia Ghosh. So Lindy, Dia, and I really became really interested in this and thought about it together over many coffee breaks and Zoom calls, and this led to this article that we all put together. Dia has since moved on from the Broad, but we really kind of definitely appreciate all her contributions to this work. Also members of Lindy's lab and my lab for many discussions around this topic. And in the MPG community, Steve McCarroll and Hillary Nuken, who kindly provided many thoughtful comments on this article.
Great. Well, thank you both for such a wonderful, starting from the beginning all the way through to such a pressing question for now. So thank you for this wonderful kickoff to the season.